I'm Llywelyn. I work in the Sheffield Neurovascular Lab. So in this lab, we're looking at sort of blood flow in the brain, how it goes wrong, and how we can target the neurovascular system to sort of treat certain different diseases. There we go. So, I mean, this is probably not really necessary for this audience, but Alzheimer's disease is characterised by the accumulation of tau and amyloid-beta in the brain. But sort of we've been alluded to once or twice already today, Bridget and Osman, that it's also associated with a decrease in cerebral blood flow. So this is sort of a really important part of it because it leads to a reduction in energy in the brain and sort of oxygen as well getting there. So you have less perfusion to your mitochondria, which are sort of supplying the energy to the neurons. So you get sort of severe neural death related to this. And also with this reduction in cerebral blood flow, you get a build-up in amyloid-beta and tau because they're not being efficiently cleared away. So it's actually like a really important part of the disease that for a long time wasn't actually looked at as much as the sort of accumulation of tau and amyloid was. And it sort of recently formed the basis of like the vascular theory of Alzheimer's disease, being that it's actually one, if not the most important part of it, at least something that really accelerates the progression of this disease. So that's what we're looking into. So the theory for vascular-based therapies is that if this decrease in cerebral blood flow is leading to sort of this acceleration of the progression of the disease, then if we can increase the cerebral blood flow, maybe we can prevent this progression, at least slow it down in some way. So yeah, the idea is that if we increase CBF, we're improving the clearance of amyloid beta and tau, so sort of preventing them from accumulating, and also we're increasing the level of glucose and oxygen getting to these mitochondria, getting to neurons, sort of helping them keep more active and healthy. And also not just the neurons, but also sort of the protective cells within the brain. So your sort of astrocytes, oligodendrocytes, if they're also sort of being more perfused with blood, then they can keep the brain healthy for longer. So in my project now, we're using a vascular-based treatment called hypercapnia. You might or might not have heard of it, but basically by slightly elevating levels of inhaled carbon dioxide, you can increase CBF without changing neural activity, which is a really important part of this sort of intervention because you don't want to be sort of messing with the neural circuitry with any treatment, but you can increase the CBF without doing that. So that's sort of an important part of this treatment program. So yeah, what we do, we literally have a chamber here. We have put the mouse cages into that, and it's very tightly controlled levels of CO2 for an hour. So I'll just describe that treatment plan in a bit more detail. So yeah, so this is the plan. So treatment plan is 1 hour daily treatments, including weekends. That was not my idea on the project, but there we go. But yeah, so 1 hour daily for 3 months using between 4.5 and 5% CO2. So we've chosen this because it doesn't cause any problems with the mice. Sort of they're completely fine with these levels. They're not perturbed in any way. But it does, we've measured using sort of a blood flow imaging that it does increase the perfusion throughout the brain consistently. So we're using this level of CO2 mixed just with normal room air. And yes, it's a 3-month treatment protocol. We're using two Alzheimer's mouse strains. So we've got the PS19 model of tauopathy. So it's got a human mutated tau expression. And we have the APP-PS1 model, which overexpresses amyloid beta. So that's our 2 models that we use. And we're splitting them. We have 3 treatment groups. So we've got the Alzheimer's group of, 2 Alzheimer's groups that are being treated. We have the Alzheimer's sham, and we have the wild type sham. So that's so we can sort of have the wild type and Alzheimer's sham. They're not put into the chamber. We just cart them into the room and put them next to the chamber. So that's sort of to assess whether just the moving them from room to room is going to affect them. And they're sort of our controls. So we also, to sort of our tests for whether our treatment are working, are mainly the behavioral memory tests. So we do them before and after we start this 3-month treatment protocol. And this is going to help us assess the cognitive decline or recovery. And we're also going to use post-mortem immunohistochemical assays to quantify brain pathology. So we're going to be looking at the so whether there's any changes in the tau buildup or the amyloid beta plaques, and also other sort of markers of vascular health within the brain, so to see whether our treatment is working. So luckily Osman's kind of already spoken through the behavior experiments that we do. I'll just refresh your memory. So we use a novel object recognition test. So this sort of relies on the idea that the mice really like attending to novel objects. So in the first instance, you habituate them to the cage. The next day, you present them with 2 of the same object, and then either 3 minutes or 24 hours later, depending on whether you're testing short-term or long-term, you present them with 1 familiar object and 1 novel object. The idea is that because they like attending to new things, if they remember they've already seen the familiar object, then they will spend more time looking at the novel object. However, if their memory is impaired, they won't remember they've seen this familiar object, and so they'll spend an equal amount of time with both of them. And then we just use this ethovision software to sort of automate our analysis, and it shows us sort of if they spent more time attending to one object or another. So we also use the Barnes maze test, which is to test spatial memory. So it's a massive arena with 20 holes around it, most of which are covered, except one which has an escape cage in it. We present an aversive stimuli, which is just basically a really bright light over the top of it, and we play white noise while it's on the arena. So that's just an incentive for the mouse. It wants to get off the arena into this escape hole. And over 2 days, we do 3 trials on each of the 2 days. And the idea is at the start, it will spend a lot of time sort of looking around different holes until it finds the escape hole. And then by the second day, the last trial, it's much more efficient at getting to the escape hole. So it's using landmarks around the room and its sort of spatial memory to go straight towards the escape hole in a cage. The escape hole cage. And then the last day, we block off the escape hole cage, and then you simply measure just how long it spends in the escape hole quadrant compared to the rest of the arena. So see whether it has still memorized it without actually having the escape cage there. So I'm just going to preface this. So we've finished 2 cohorts of the Tau study, and last week we finished 2 cohorts of the APP study. So this is very preliminary data, and unfortunately due to the randomization of it, it's one of the groups, the Tau sham group, is an N of 1. So not really making any conclusions from this data, but this is sort of just a quick look at what we're showing and how we're going to be presenting it. But this is the results of novel object recognition for the short-term memory. So that's when there's a 3-minute delay between these sorts of, when they've seen the familiar objects and then the novel object. And the preference index, the important thing here. So if it was at 50%, then it would have spent an equal amount of time with both of the 2 objects. Anything over 50% means that it has at least some memory of this novel object. And just the one thing I'd sort of point you to is, looks at in this Tau treated, so this is our Tau mice, they've had the treatment, it seems quite similar, so maybe, the Tau sham is showing a different difference, so we can't really make any conclusions, but at the moment it looks like maybe the Tau treated is the same before and after our treatment, so that's maybe stopping the progression of the disease. So this is our long-term memory, so this is a 24-hour gap, and what's interesting about this is you'll actually see quite a lot of it falls below the 50% mark. So what that means is actually spending more time looking at the familiar object rather than the novel object. So what we interpreted this as is it's not worked, basically. This, over 24 hours, it has none of the categories of mice. We've combined all of these and all the others that we've done together, and we've shown that statistically it's not different from 50%. So basically, they're not remembering the objects over 24 hours. So we're actually excluded that from the study for the rest of the study. So sort of going to the point that it doesn't always work as you planned out to. And then this is sort of the Barnes maze findings. So have the latency to locate the escape hole. So that's literally just how long until it finds it the first time that it finds the escape hole. And this is the time to actually escape, so when it climbs down. And sort of pre-treatment, you can see that they're all seemingly learning, so it's going down. Again, this N of 1 looks a little bit odd, but for the larger groups, it's all over the multiple trials. They're sort of learning where the escape hole is, getting quicker and quicker each time, which is the idea behind it. And then this is after the treatment, so 3 months later. And again, if I was to hand wave your conclusion, you could say that the Tau treated time to escape is lying between our wild type sham and our Tau sham. So that would be, our hypothesis would be that it falls there, but again, we're going to have to increase N numbers before we make any conclusions from this. So our hypothesis for these studies are that the CO2 treatment is going to improve the memory in both the Tau and the amyloid mouse models, and sort of compared to both the Tau treated, and also hopefully bring it back to the wild type sham sort of level would be the ideal finding. Also that our immunohistochemical analysis is going to show that this treatment has sort of improved the brain pathology, sort of reduced maybe the plaque load and the Tau buildup. And looking further ahead, so if our treatment is shown to work, then it provides really good evidence that increasing blood flow can improve the prognosis of Alzheimer's disease. So it's sort of showing there should be a lot more emphasis on vascular-based therapies. Now, this maybe not this particular therapy in humans. I mean, elevating CO2 levels can be known to cause lightheadedness and slight delirium in humans over a certain level. So it might be more difficult. I mean, it could be fine-tuned for humans, but it might not be the first port of call. But it's just a really good example of like a proof of principle for using these therapies to treat Alzheimer's disease. And thank you very much for listening. I'd just like to thank everyone in my lab and Claire and Jason, who are the PIs on this grant. And yeah, any questions?
What histology markers are you going to look at?
Ooh, so we're currently discussing it. So we're using, we've got 88 to stain for tau, and we're using methoxyXO5 to stain for amyloid. Then we're looking, I think we're going to look at astrocytes, so like GFAP, and we're going to be looking at some of the vascular markers, so like endothelial cells and stuff like that. And maybe also we're interested in nNOS. There's sort of an idea that nNOS degradation in Alzheimer's disease is correlated with the sort of CBF decrease that we see. So we're also going to be staining for nNOS. Basically everything we can look at.
Do you look at any gene changes to correlate with the...
Not currently planned to, but I mean we could do. Would there be any gene changes you suggest?
No, I mean if you're looking for some active changes, would you expect to see them with the long term?
Interesting. Yeah, that's a good idea.